Well, we'll go ahead and get started. Good afternoon, good evening, and good morning to everyone, wherever you might be. Welcome to the next installment in Sino Biological's sponsored and hosted Lock and Key webinar series. I am Rob Burgess, and I head up business development for Sino Biological. We have an extra special seminar that's going to be given by a stellar scientist and entrepreneur today. And in a minute, I will introduce that individual to you. Before I do that, though, I just want to mention one very brief housekeeping issue, and that is I ask everybody to type your questions into the chat box, and we will withhold all those questions until the end of the seminar, and then I'll walk through those questions and get to as many of them verbally as I can. Any questions that we don't get to, then we can have answered offline by email, if that's all right. One other thing is please put in the chat box where you're from and who you are. We always like to see worldwide where the attendance is coming from. I'd really appreciate that. And so with that, without further ado, it is my pleasure and honor to introduce to everybody on the webinar, Dr. Jacob Glanville, who will be giving our seminar today. Dr. Glanville is a serial entrepreneur and a computational immunoengineer. He built and sold his first company, Distributed Bio, from founding in March of 2012 to Charles River Laboratories in December of 2020, in which it was sold. During that period, he developed the core business model, the research teams, and all of the technologies that really enabled Distributed Bio to become profitable without any further investment. As part of the acquisition agreement, he founded Centivax, which is what we're going to hear about today, and spun out his assets into COVID-19-based therapeutics, broad spectrum vaccines as well, anti-venom antibodies, anti-wound pathogen antibodies, anti-CXCR5 autoimmunotherapeutics, and blood-brain barrier translation technologies into Centivax, where Dr. Glanville is now the CEO. Dr. Glanville obtained his bachelor's from UC Berkeley, and he obtained his doctorate from Stanford under the advisement of Dr. Mark Davis in computational systems immunology. And one other very interesting thing about Dr. Glanville is he was recently featured and interviewed on the Netflix documentation series, Pandemic: How to Prevent an Outbreak. So I encourage everybody to watch that very interesting documentary if you have a subscription to Netflix. And so the title of Dr. Glanville's talk today is Universal Vaccines, Universal Anti-venom, and Broadly Neutralizing Antibodies. And with that, Dr. Glanville, I will turn the screen over to you.
Thank you very much, Dr. Burgess. I want to thank Sino Biological for hosting me, but also, as you'll see throughout this talk, for an extremely productive relationship that we've had that's affected all of these projects since 2015. I'd also like to thank all of you for coming and joining me. I hope to go through some technology, some background, and talk a little bit about the future of the application of broadly neutralizing antibody biology that's of interest to you, hopefully of practical utility to you, and hopefully spawn some thought and some questions. So you've already heard a bit about me, so I can skip my self-intro. That's great. And I'm going to go jump right into the topic at hand. I think, as you heard, if you have questions, put them in the chat and we'll get to them towards the end once I've concluded. So let me introduce the company very briefly. So we're located in South San Francisco at the JLabs incubator. We have animal facilities that we use from Charles River Laboratories nearby. It's a team of 15 people, as well as I'm showing our advisors and our board members. It's deliberately set up so that every level of the company is experts in their fields, from regulatory to manufacturing to immunology, computational engineering, and so forth. The company is supported by the Gates Foundation, the Navy, the Army, the NIH, and a number of other great organizations that believe, like we do, that broad spectrum vaccines and broadly neutralizing antibodies are an important part of being able to ensure global health security. And as was mentioned, because of the nature of our work, we are featured relatively frequently on national media and other sources, and we try to do what we can, obviously, to promote our own work and our technologies, but also to be useful during the course of the pandemic or when there's questions about new pathogens that arise. I think we're in this place because infectious disease is humanity's greatest and most ancient enemy. It kills more than every war combined. It has affected us since the beginning of time, and every one of us confronts it within months of birth. And so this is a common problem, and we feel that we live in a modern era, and yet we are still constantly plagued by many pathogens. And this is part of the work that we're doing and the purpose of the company is to try to address that. And I'd like to share with you some cool new biology that's emerged in the last 20 years and our work in that space that's enabling us to begin tackling some of these previously very challenging pathogenic and other targets. So the core problem here is that mutations cause our vaccines and our antibody therapeutics to fail. So I love vaccines. They're one of the greatest medical advances since sanitation and fire. But even though we successfully eradicated in 1980 the smallpox virus, something that had left scars on the face of the pharaoh, and yet we were able to eliminate it, we have struggled to do the same since. And in some cases, there's political and global challenges, but the other challenge is that our current vaccine technology, our current antibody approaches deteriorate against rapidly mutating pathogens. And we've all experienced this because of the pandemic with the rapid mutation of the coronavirus. We've kind of gotten used to it. We shouldn't for the influenza virus. And it's why we don't have working vaccines for a number of additional pathogens. And so that's the area I'd like to be able to focus on today. Some novel biology that lets us address that and some bioengineering to get there. I'd like to start with just a high-level overview of the concept of a broadly neutralizing antibody. So for a long time, we thought, yeah, there's nothing you can do. These pathogens are going to mutate too much. It is what it is. And then in the 2000s, like 2000, 2004, 2006, there were a set of these very beautiful papers that came out that said, hey, wait a second. And influenza and HIV, and in fact, any virus that you look at, it turns out some lucky devils are producing antibodies. They're recognizing these super conserved sites that even though the virus is generally mutating everywhere it can, there's certain sites it cannot mutate. And if you make antibodies against those sites, you enjoy ultra broad protection against many future and variant versions of those pathogens. I mean, on one hand, that was very exciting because that said, well, therefore, if we can get our vaccines to make those kinds of antibodies, or if we can engineer them ourselves, you can come up with great new broad spectrum or universal vaccines or broad spectrum therapies. But the challenge was getting the body to do it. And that's what I want to talk about today. In order to understand how broadly neutralizing antibodies work, I want to tell you a little story. Back in World War II, the British were wanting to try to reinforce, put more metal on their planes. And they started collecting the data from all the planes that came back. They started recording where all the bullet holes were. And those are all those red dots. And so as they gathered it together, they came up with this picture. And someone's like, okay, well, it seems like the wings are getting shot a lot in the area in the middle a little bit. So those are the areas we should reinforce with metal. And then someone's like, no, you idiot. It's the spots where you don't see bullet holes. Those are the parts you need to reinforce because those are the ones where if the plane gets hit there, the plane doesn't come back. And I want you to look at that again because this is exactly how biology works. That biology can mutate just about anywhere. That's what the virus is doing. But if you see a certain site the biology does not mutate, it's because the mutations happen, but the virus was no longer viable when it mutated there. And so those sites that don't mutate are critical for the function of the virus. And if you can make an antibody against them, you enjoy broad protection. This is illustrating it now with the influenza virus. I'm showing as the virus evolves, you can see that heat map of the original sites in yellow and light green start shifting. More and more sites are mutated, but there's a site at the top that's hard to mutate there in the middle. And on the bottom, the stem, there's a site which is super conserved. And the virus could never mutate that site because if it mutates that site, the virus dies. It can no longer infect anymore. And if you see on the right, that's exactly where those broadly neutralizing antibodies bind. So that's what they are. They're antibodies that happen to bind a very precise epitope, recognizing the exact spot that the virus cannot mutate, sticking the virus between a rock and a hard place of not being able to mutate that site to escape the antibody because it becomes no longer viable. The challenge is in generating them, to getting the body to generate them and to engineer them. So I'm going to start, there's a couple topic areas I'm going to go over, and I'm going to start by talking about the vaccine application. After that, I'll talk about inducing them in vitro and some unique opportunities to elicit them against anti-venom outside of the area of viruses. The pictures I'm showing you here on the left, this is how we generate our very large libraries of diverse viruses for neutralization assays. They're developed in eggs. It's a very scary set of eggs when they're ready to harvest, because you've got 90 years of virus growing there. And then on the right, I worked with pigs for the initial set of research. I'm going to show you pig and ferret data. Pigs are a great model for flu because they get the flu. There's a market, $180 million per year market for flu vaccines for pigs. And pigs also need a broad spectrum vaccine that works better. So that was at our animal facility in Guatemala, where some of the early work was done. You'll see that on the Netflix documentary series. Although these days our research is predominantly done at the University of Georgia, Auburn University, when it comes to pigs and ferrets. We work with academic partners. Okay, so why vaccines? Like I mentioned, vaccines have been a remarkably successful technology that has changed modern life. They are a massive blockbusters and growing area of the therapeutic landscape. And they're great medicine because they don't cost that much, and you can give them to a lot of people, and it avoids getting sick in the first place, which is generally a better strategy in medicine. This has also been a great place for a number of new powerful IPOs that have come up recently. So it's fertile ground for a company. The question, of course, is like, well, then why do you need you? Why? It seems like this industry is doing pretty well. And it's for the problem that I mentioned, that for all the success, our current vaccine technologies do not work particularly well against mutating viruses, rapidly mutating viruses. This is flu. The red line is how well the flu shot works every year. So it ranges from about 30 to 60% effective. And in some years we miss pretty bad and it's down to 10% effective. And that's because we have to make a guess almost 8 months before the season over what components are going to go in the vaccines. And it's too risky to pick the most recent components because of manufacturing risk delays. So people compromise, and they sometimes leave in older components in those vaccines. And that means a lot of your antibodies against what's in the vaccine is not, those sites have changed on the virus you're actually infected with. And so I've highlighted above where we actually should be. A good vaccine should be 85 to 95% effective. And the missing delta here is due to mutation. So since those broadly neutralizing antibodies papers came out, a bunch of people tried a bunch of cool things to try to get the body to target those conserved sites. And it turned out that was pretty difficult to do. And it was kind of frustrating, and people were struggling to understand why these methods were failing. And part of the challenge was that the immune system is a big, complicated black box, and it was impossible at the time to go in. We did lack the technologies to try to understand why were we missing? Why was that conserved site always there, and yet we weren't targeting it? And some of you may have heard some, I would say, colorful theories that I'm somewhat skeptical of about some of the arguments for why we're missing those epitopes. I think saying maybe they resemble self proteins that are being deselected against, or other spooky arguments that I don't need to say that they're wrong. What I can say is that they're not necessary, because there was a misunderstanding about the nature of how adaptive immune systems target surfaces. I'll just mention it because it's the elephant in the room. So of course, people tried after the success of the RNA vaccines to apply that same technology. Like, maybe we just put RNA on it and we'll solve the flu problem. Of course, it did not work and how could it? Because you're still putting the wrong RNA for the wrong strain in the vaccine compared to what you're exposed to that flu season. And so it didn't work any better than the seasonal flu, but it had more side effects. Look, I take the RNA vaccines. I'm glad I did. I'm very appreciative of those scientists, but they do have more side effects, and that's harder to argue bringing a vaccine with more side effects that doesn't work any better into the flu space. So what I was interested in for the last 15 years was why we miss. Like, what was the real reason why it was so hard to get the immune system to focus on these sites? I felt that we were lacking some engineering principles that, if we had them, could help us guide towards engineering better vaccines. And so I and others were fortunate that right around this time, right around 2008, 2009, high throughput sequencing instruments and other high throughput data acquisition instruments started coming online. And while I was at Pfizer, I began applying them to the human immune system. And so as a consequence, with my work and others, we've gained a lot more insights into why it is that we miss with vaccines. On the left, to summarize a bunch of work, after you get a flu shot, there's about 1,000 unique B cell lineages that wake up, and about 10% of them are producing the antibodies that protect you. So you get about 100 antibodies after a vaccine, different types of antibodies. That was much less than people thought, but that's the numbers that we have. In the middle, you go, okay, well, how many sites are there on the hemagglutinin, on the spike protein? If it's less than 100, you should be hitting them all the time. Why are we missing? And it turns out that there's a lot more than 100. By using cloud-based massive protein-protein docking experiments of every possible structure for human antibodies against every possible surface on hemagglutinin, we come up with an estimate of more like a few billion possible epitopes. And so your 100 antibodies are just barely sampling the universe of possible binding sites. And what that means is, okay, if you're not hitting them all, then it really matters what's the distribution of conservation. And so on the right is our analysis there. That can be easily assessed by taking the sequencing data that we have back to 1918, year after year, and compare it to those billions of epitopes and ask, what's the proportion of them that are the universal epitopes compared to the ones that are not? And this is really where the rubber hits the road on the problem. You have about 80% of those sites are not conserved at all. They're going to change in the next 1 to 3 years. And so, 4/5 of your titer is susceptible to rapid change. 19% are somewhat conserved as to within subtypes. So you're going to have some titer that has some residual long-term staying power, but that's still, your titer is going to go down 4/5, and then the other titer is going to deteriorate over time. And those really cool, the sexy bnAbs that people published on, the reason they weren't in every person and the reason why it took effort for them to go find them is that those things are less than 1%. And the really good ones are like 1 in 10,000 to 1 in a million. So they're very rare. They're always there, but they're hidden in this ocean of nonsense. And that's why we miss. And that's why those technologies in the first 15 years since the discovery of bnAbs really struggled to focus the immune response because there's too many wrong answers and they're floating around in the middle and being missed. So our technology, our patented technology to address this works as follows. We say, we're going to take some HA antigens, and this approach in the patent covers, our patent covers any pathogen, but I'll talk about flu right now. We take a bunch of HAs that are from the last 100 years. They're all really different from each other. So let's say you took 10 of them. You then mix them and you dilute them so that there's not enough of each individual HA. Each HA by itself is not enough dose to create an immune response. And that means that B cells that recognize that particular HA strain, they don't get enough vaccine to activate. Whereas a B cell that recognizes a conserved site across all 10 components, it's going to get 10 times the dose. And so it's going to be above the threshold. And that through that mechanism of taking diverse members and then diluting them below the individual threshold, what you're doing is you're selectively delivering the vaccine just to the B cells capable of producing broadly neutralizing antibodies. So they're no longer being outcompeted by the overwhelming number of wrong answers, and your entire population of your 100 antibodies are all broadly neutralizing antibodies. So that's the technology. I want to show you the data and the results that we've been getting and why we've been building up so much momentum around this. This is just to visualize that again. So I'm showing you now as you lower the dose, so you have each antigen's at half a necessary dose, a third, a fourth, a sixth in the mixture, you increasingly focus the immune response on the most conserved sites. We call this technology concentration conservation coupling. And what you're doing is creating a linear relationship of the dose a B cell receives by the breadth of the epitope they recognize. This was one of our first studies. This is in pigs. At the Guatemalan facility. Most of the data I'm going to show you is from the United States laboratories. This is part of the data that got us awarded the Gates Foundation Grand Challenge in the Pandemic Threat Award. On the left are 7 pigs that received squalene and no HAs. By the way, all the HAs you're seeing here, these entire things, Sino is always our partner for all of this work. So got the original ones from them. And I could not have started this study if it was not for Sino having this repository of HAs, because early on, no one was interested in broad spectrum vaccines back in 2015. And so they had this beautiful panel of HAs. And so I was able to order a series from them and start running these experiments. And so thank you, Sino, for doing that. It's a community service and we're super appreciative. And all the HAs, I always work with Sino for all the HAs. They do fantastic work. On the left, you're seeing the pigs that received just squalene. They don't respond to any of that panel of HAs that goes from 1918 up to modern times. Pigs in the middle, they respond predominantly to the 2 things with the stars. We vaccinated them with 2 HAs, like a standard bivalent flu shot for pigs. And they respond to a couple years around it, but that's about it. That's the whole flu problem. And on the right are the pigs that received our low dose mixture. And they respond to a century of influenza, all 5 pandemics of the last 120 years, and a whole bunch of things that were not in the vaccine. So we induced heterologous outspreading, which is what is anticipated for the technology. The bars, which are still active but are lower, if you look at them, they're things like H7N7s and H5N1. So we're actually crossing out to the exotic subtypes that normally only infect birds, but less so than the things that humans care about, to be fair. So then we wanted to run these live challenge studies where we spray animals in the face with future virus, to see if they get sick or not. So the way we set these experiments up is we pretended it was 2007. And we said, we're going to use components from 1918 up to 2007, but nothing after that. And we're going to compare those to a different cohort of pigs that receive, or ferrets that just receive a 2007 flu shot. And the reason we do that is that we can then test those animals. We can test their serum by hemagglutination inhibition and microneutralization assays against future viruses out 13 years into the future to see how does their immunity protect them for the decade to come. We can spray them in the face with virus from the pandemic 2009 antigenic shift virus. That's like the Omicron of influenza. From the 2012 swine adapted H3N2 at 3000 and H3N2 from 2017. So we can do these future challenge experiments. And that's the critical way to set up these kinds of experiments. So with respect to the hemagglutination inhibition assay, and this is an important one. This is what the FDA wants to see to approve your drug. It's the correlate of protection, which is standard. You can see the bivalent and the trivalent for the pigs and the ferrets, respectively. They don't do a whole lot against most of these panel. That's the whole problem with flu. That's why we remake it every year. You can see the pigs basically entirely fail as you go into the future. The ferrets, they do pretty well on HABs. They're much more conserved, so they are able to hit future 2007 to 19 HAB. But HAB also is the most conserved, and it doesn't cause pandemics, and it has the least severe disease. You see they really struggle with H1N1s, and they're struggling, doing a little better with H3N2s. When you shift over to our vaccine, in the middle, on the same adjuvant, our vaccine is producing this ultra broad immune response in the pigs and in the ferrets compared to the standard. And then when we change adjuvants, we've got a formulation right now for pigs that just takes our technology, and it makes it convenient. You can do a single shot. You could boost them if you want, but a single shot sufficient that gets this super, super high titers on everything we've tested out. I think at this point, we've done 13 years into the future. And similarly, we get these ultra broad responses in the ferrets, and there's multiple adjuvants we can work with there. So we're talking to veterinary partners already to create a broad spectrum, a universal vaccine for flu for pigs, which is great because pigs are the recombination species where bird viruses get in and mix with pig and human viruses and cause pandemics. And so getting them away from the pigs is not just saving our bacon, it will protect us as well. And for ferrets, that's our critical model to get into the human studies that we're entering manufacturing for that purpose. We did live challenges, as I mentioned. We are the only technology that I'm aware of that was able to take a pre-2009, so 2007 formulation, and then the animals so immunized were protected from a pandemic antigen shift 2009 challenge. And you can see that here. They are almost entirely protected from any weight loss, almost entirely protected from any symptoms. And similarly, out to 2017, 10 years in the future, we also continue to provide protection for those animals for an H1 and an H3. The pigs challenge strain was relatively mild, so we do protect them from, but it's like there was not much more symptoms when it came to the H3N2 2012 in the mock. But where we saw a huge difference is that when we went into to look at the viral titers, we found undetectable virus in most of the regions that we tested in most of the animals. So nasal turbinates, trachea, left lung, right lung. We end up seeing 100 to 1,000-fold reduction in virus. So this not only means that this is protecting the animal very vigorously from being able to be colonized by the virus. We also suspect this means that they're no longer able to transmit, and we have studies set up right now to validate that. So this, I know this looks a little bit like a Druid's cookbook, but this is a way of comparing the breadth. And by every possible method that we have to compare breadth, we have defined what breadth is, this new epoch of broad spectrum. So this is by binding assays, by hemagglutination inhibition assays, by neutralization assays, serum ELISAs on pigs and ferrets against H1s, H3s. We also go out to H5s, H7s, and so forth. So, and this is, great and powerful validation of this approach of focusing the entire immune response on the parts of the virus that the virus cannot change. There's a manufacturing advantage here I want to touch on briefly. So right now we have to do this crazy thing on the left that I mentioned, where you have to go make a new vaccine every year, and in the northern and separately in the southern hemisphere. There's a risk there, because if you go, say, at the end of March, go, all right, guys, let's start making the new flu shot. You have to go manufacture the damn thing, and you have to get it approved, and you have to start producing enough batches to be able to go serve that market. And there are delays in manufacturing sometimes, which can be very risky for the organization. And as a consequence, the FDA, the CDC, the World Health Organization, they compromise on letting less well-matched vaccines go through so they don't have to remake all the components. It's also just very risky business. So in contrast, our vaccine doesn't need to be updated every year. And that means that you can just manufacture it and stockpile it. You can you can serve the northern and the southern hemisphere, and you can attack those markets right when they open to sell your vaccine at a premium and be confident you can fill all your orders in time. So it's the same way that normal therapeutic business should be run. We can bring that to the vaccine space for influenza. This is our manufacturing strategy. So we do, again, we've worked with the HA so far with Sino Biological. We're working with them for vetting reasons around our CHO work right now. We're going to another partner to produce the GMP cell lines. Although if Sino opens that up, I would work with Sino, because I've just been consistently very happy with that group. They're not paying me to say any of this. I literally, the success of my company, I attribute to the excellence of their work in some regard. And so I'm very appreciative. But that's our strategy. The beauty of this technique is that we don't have to use weird expression systems. We can use CHO. It's super commoditized. We can grow these massive batches, and we can produce a global supply in a reducible, consistent, and stable fashion. We figured out there's a couple of things that probably don't warrant discussion here, but there's strategies around fill finish and batch release that have also been sorted out. One of the other advantages of our technology is that with normal flu shots, or any virus for that matter, you're kind of like stuck with whatever the antigen is. So if the antigen sucks and you're like, this thing aggregates, it doesn't express well, it's not thermostable, it falls apart, it has poor shelf life, you kind of just like, tough, that's what we're doing. Like, we're injecting A to produce a response to A. Where our technology creates this opportunity to do better than that. Our technology requires that the components are all quite diverse from each other. It doesn't actually matter so much which exact member it is, as long as they're all sort of a diverse snapshot of the evolution of the viral space. And what that means is that we can go strategically pick a series of antigens and compare and pick the ones that are better expressing and more stable and have better shelf life or more compatible with our adjuvants, as long as they're diverse from each other. And this has been another place where we've been working with Sino on this. We develop large panels, we do a series of screening, and this enables us to produce a product which is going to be more shelf stable, and it's going to be more reliably efficacious over a long period of time. And we can manufacture a global supply better at a lower cost of goods. And this is a unique advantage of our technology, because we have some flexibility over the components we choose to add into the vaccine, and therefore we pick the better ones. So far, I talked about flu. We are also working on the coronavirus and HIV as our other two target areas. Again, we work with Sino quite a bit on these antigen panels. They have reagents, they have antigen panels, and we do the custom engineering work, and they're producing these proteins. So it's just been a joy to work with. And there are 17 other pathogens that we're looking at next once we get these first 3 into clinic. And so my intention is to keep working with Sino for those as we go forward. All right, so I just want to touch upon one final point before we go to the next topic, and that is the reagents for broad spectrum vaccines. So when you're trying to make a broad spectrum vaccine, then you have an additional challenge, not only on the experimentals, the way I described it, but also how do you set up your assays to make sure your assays are able to successfully run the same way, given a panel of very diverse viruses that might span a century in our case. And I want to describe to you why that matters. So lots of people use this neutralization assay, where you basically take some cells, you put the virus on it, and the virus will infect the cells, and it starts expressing the nuclear protein, NP. Then you detect it with an antibody. And then you can take the serum of an animal, mix it in there too, and go, okay, does it block the virus from infecting the cells? So that assay seems well and fine, but imagine if you now have viruses from the last 100 years. People often don't think about the reagents, but how reliably do you think your antibody, your anti-MP antibody, is going to detect the MP from the century? And I've heard immunologists be like, oh, MP is conserved. I'm like, that's bullshit. Excuse my French. This is an analysis of the polymorphism level of the MP protein over the last century. You can see there's a considerable amount of variation there. And then even if it wasn't varied, the antibody quality matters for a reagent with respect to the signal, your background, and other characteristics. So we conducted a study where we tested 12 different commercially available anti-MP antibodies as we were setting up these assays. And this was Sarah Ives who conducted this work. I'll give you a hint. It's going to be the Sino antibodies. Number 8 ultimately was the winner, which was great. I'll show you the reasons why it actually mattered. So we ran microneutralization max anti-MPODs across this entire panel, a diverse set of viruses. We compared it to 8 viruses, H1s and H3s, and we figured out what the apparent max OD that was detectable. And you can see some of the antibodies just completely failed out. They were non-active. I'm on a paper with Bradbury discussing some of the problem with reagent antibodies. And for the ones that did, there was also considerable variation on their breadth of detection. Some of them worked way better on one subtype than another. And you can see some that were missing and are doing better or worse at reliably detecting all the components. I'll highlight AB8 over there. That's the lead clone from Sino that we work with that has the most consistent active ability to detect all the viruses. Next thing we would do is look for background signal. And again, there were 2 antibodies there that we considered unacceptable for further use. One of them is totally useless, and the other one's not good. AB8's under our threshold, so we're happy with it. And then we were performing titer determinations. Our technical replicates, Sarah's replicates were excellent here. This is totally a difference of these antibodies and their breadth of epitope and their ability to equally recognize MP from different viruses. And you can see that matters because you can end up getting a half log to a log of variation and therefore get false readings in your studies on how good of neutralization you think you're getting, where it's really an artifact that your antibody doesn't perform as well depending on the virus. These are things that don't matter so much if you're just working on one virus because everything's relative to each other. So you might be off in the absolute, but it doesn't matter in the relative. But when you start trying to create broadly neutralizing antibodies, it matters a lot. And I wanted to be able to emphasize that point. So first off, thanks for having a great reagent. And it's important to think about that when you're trying to work on broad spectrum therapeutics. All right, so let's shift gears a little bit to inducing breadth. So far, I talked about in vivo induction of breadth. What I want to talk about now was a technology question where we asked, could we take SARS-CoV-1 antibodies and then evolve them to become broadly neutralizing against SARS-CoV-2 as well? This is just coronavirus pinata we had the other day. So I think everyone knows how this works. The coronavirus spike has this thing called the RBD on its spike. And if you get an antibody blocking that RBD, it can't go bind to the human ACE2 receptor. So it's been a dominant target for vaccines and for monoclonal therapeutics. So what I did at the beginning of the pandemic is I went out and I found a bunch of antibodies that were known neutralizing antibodies against SARS-CoV-1. This is back when we didn't know anything about SARS-CoV-2 yet. And I said, I bet we could take those antibodies and I bet we could evolve some of them to be able to be neutralizers against CoV-2. We already know they have the function, the activity, the surface has just changed a bit. So let's change the other side. Let's change the antibody surface to adapt them. And it was an important question because it lets us ask, how malleable are existing antibodies we've already discovered against a cousin of a new pandemic virus to follow? So these are the 5 we chose. Some came from healthy humans, some came from immunized mice, some came from SARS patients. They all came from SARS, almost 20 years before, or 15 years before, whatever. And they also hit multiple different epitopes because these are all like crystal structures. So I used a computational method for optimizing the antibody library to strategically explore variation across all 6 CDRs. And I also did some engineering to try to optimize the developability characteristics. I wanted to adapt them to SARS-CoV-2 and also optimize them to be super good drugs. This is, I won't spend time on it. This is basically our workflow. It's a bunch of fun devices so you can work pretty quickly. We ended up doing the panning process in 5 days. The discovery was super fast. In this, we had our lead molecules by March 1st of 2020. And the answer was all 5 cases, we were able to find antibodies that crossed over to CoV-2 from the CoV-1 start antibodies. So this is generally it is possible to take an antibody and shift it over, although in some cases they jumped epitopes. And in one case, the 6MB6, we lost affinity when we converted to IgG. But we got this distribution. We're getting down to picomolar binders in some of these cases. these great looking antibodies that we're able to rapidly convert. So the answer is generally yes, you can in vitro evolve a CoV1 antibody to be able to adapt to CoV2. And there's implications here for pandemic preparedness in the future. Many of them kept their same epitope, although we did find new antibodies that were actually jumping epitopes. So even though they looked very similar to the previous antibody, we had changed enough residues that they'd found a new way to go bind the surface. And down below, I've highlighted the set of mutations that were accumulated from the lead CoV1 antibody to adapting them to be a CoV2. Two of these became bnAbs. They hit CoV1 and CoV2, and we have some molecules that we continue to track that went through Delta and Omicron and continue to bind very broadly. So this is also a method for essentially refining towards universality. We worked with, this is the middle of the pandemic, 6 laboratories all independently confirmed the neutralization capability of these antibodies against the novel coronavirus and a number of subvariants. They were active in vivo, so they protected Syrian hamsters at 2 different facilities from a couple different formats of prophylaxis and preventative prophylaxis and treatment models. I guess here's all the figures from the paper. In the interest of time, I'll skip it. But the punchline is you could take these CoV1 antibodies that were known neutralizers, you can convert them and they preserve their function and their epitope, and they carry out, they're potent in the future, which is a super fast process. You can do that much faster than you can go wait for some convalescent person to show up and take their cells, and hopefully it's a good one, which you've noticed that was a very hit or miss proposition, and most of those antibodies have failed. Although I'll be fair, our antibody that we took the furthest here, it also got damaged by Omicron. Protected the animals, all those good things. One of the other advantages I mentioned, I also was optimizing developability, and this was kind of a big one. So as long as you're building a library to explore variation, you can also use that library to explore optimizing the therapeutic properties. So on top, you can see where the box is where the original antibody was in terms of humanness, and then the leads are shown in all the little dots. And you can see we basically made these antibodies much more human in almost all cases. One of them was already pretty human, 2DD8. And then we dramatically enhanced their thermostability and their aggregation resistance. And the effect of it, and we lowered the viscosity. And the effect of that is that we GMP'd this molecule at 254 mg per mil. It was ultra thermostable and was able to tolerate really high concentration formulation delivery. And that's exciting because there's certain types of medicines we'd rather get them as a subcutaneous injection or an IM injection and be able to walk out of the medical center. So it's an outpatient procedure, or there's some medicines you want to give out in the field. And for military reasons, for local clinic reasons, there's a number of other reasons. And so engineering antibodies, I think we can take them much further than people think we can, and you can do it in this integrated fashion. I've also mentioned that we've done this on HIV. I basically never bought the argument that antibodies against HIV that were neutralizing had to be these super screwed up, highly mutated ones. And I was like, no, man, that's just because they're getting those antibodies from an HIV patient who's been infected for some years before the antibody pops out. So it's a function of the history. It is not a requirement. And we proved that by working with Peter Kim at Stanford, where we pulled out these great bnAbs that were almost entirely germline. They had some mutations in the CDRs only, but the frameworks were nice and clean. So these have the advantage over existing HIV bnAbs. You can express them really well. They're very thermostable. They're not like disasters to work with. That is not a requirement. That is an artifact of the history of how bnAbs were traditionally collected. All right, so I think I have a few more minutes left, and I wanted to tell you this story because it's really cool, and it illustrates the power of bnAbs are not limited to viruses, and that is the story of anti-venom broadly neutralizing antibodies. That's me holding up some toxin from a snake. So I was interested, of course, in what does it take for a human to produce broadly neutralizing antibodies, and I was particularly interested in the snake antivenom because there's 600 species of snake around the world that are venomous, and each one's injecting you with maybe 10 or 20, sometimes up to 70 toxins. So it seems like a really big problem to create a single drug against. And in fact, right now, what they have is lots of antivenoms that are made in horse and sheep that treat one snake or a limited set of snakes. And it's a fractured market. And most of the people who need this don't have a lot of money. They're going to be farm workers and their children. So I found this guy. I was like, if we could get broadly neutralizing antibodies against the common toxins found in all snakes, then we wouldn't actually need that many antibodies. The product would be more uniform and reliably effective. it could be fully human, which means it could be injected in field setting and be much safer to the patient. And so for all these reasons, I was, obsessed with bnAbs, and I was looking for someone who'd been bit a couple times by a snake. I was hoping for like a clumsy snake researcher. And instead, I find this remarkable guy named Tim Freedy, who for 17 years and 9 months had 856 escalating doses of exposure to like 16 different species of snake from around the world, cobras and taipans and mambas and rattlesnakes and a whole list. They're all down there. And he documented it in exquisite detail the entire time. This was his life's work. So I'm seeing some of the data, a snapshot of this massive 20-year study he ran against himself. And so I contacted him and said, hey, could I get a little bit of your blood? I think you may have done something remarkable. I think by cycling between these snakes, I think you may have selected for broadly neutralizing antibodies, which could be the basis of a universal antivenom. And so he agreed. The figures I'm showing you are a paper which is currently in review. You can go up to bioRxiv. You could read it right now. Just search my name, Tim Freedy, Peter Kwong. So first thing I did is I tested his blood, and sure enough, on the left, he had blazingly high signal serum ELISA against 4 different genera of snake compared to healthy controls. And then on the right, I looked at the mutational level, somatic hypermutation of his antibody repertoire. It was more elevated than normal humans. And when I looked at the subset of his repertoire that was against anything in venom, it was even much more elevated than that. And the broadly neutralizing antibodies I'll tell you about, those were the most mutated in his repertoire, which makes sense if every time he was immunizing, those things were getting stimulation to mutate over and over again. And he was doing it for almost 18 years. So this was our process. We took 20 mils of his blood, we built a phage display library, and we produced recombinant toxins, and we panned the library, changing out the homologue of the toxin between each panning round. After that, in principle, that would select for the broadly neutralizing antibodies. We would isolate them and characterize them. And we would do that for each of the major toxins. We chose to start with alpha neurotoxin. This is the major toxin, long neurotoxin in snakes. We like it because it targets the nicotinic acetylcholine receptor here on the left, which is ultra conserved all the way out to fish. So it has a single target across all snakes that's hitting basically the same surface. And on the right, you can see the toxin. You can see that imposes upon the toxin in that region in red. There's part of it that's super conserved. It can't change part of its interface, or it's going to no longer bind its target. We're back to that plain example again. And so I thought this would be a great place to look for bnAbs. So we did the search. We found a set of bnAbs. There's a couple of main clades that popped up, and the dominant one, we have an antibody called D9 that I'll show you the rest of the data on that came out of that clade. So D9 was ultra potent. It was a low picomolar binder, so 74 picomolar, 37 picomolar. I think the worst we saw was 490 picomolar against extremely divergent versions of the neurotoxin. These things are like 57 to 61% identity, I guess 64% identity at the amino acid level, like so diverse. And yet this thing was binding them consistently in this ultra potent fashion. We developed an assay to allow us to use biolayer interferometry to look at whole venom. And we screened a panel of 20 elapids, and we found that it bound all of them except for 2. One of them is known not to contain long neurotoxin. The other one, I can't find the sequence in the database, but we haven't confirmed that it's absent. But we suspect it is also absent. But the other 18, it very potently bound them all in a similar manner. In the interest of time, I'll skip this. This is just that we validated it by doing immunoprecipitation pull down. This is work at Peter Kwong's group by Baoshan. And then we mass spec those individual bands that came out of the expected neurotoxin length. And sure enough, those contained also neurotoxin. So we've validated this in every direction. This thing is very precisely, specifically, and with ultra high affinity, binding all the neurotoxins from all snakes that bear that toxin. So the question was, how did it bind? And so Peter Kwong's group and a guy named Raffaello there developed a crystal structure of this antibody in the context of that toxin from 4 different genera. Like, this is a tremendous amount of evolutionary distance: the taipans, the cobras, the mambas, and the kraits. You can see it binds the same way in all the cases. This is the power of a broadly neutralizing antibody. And when you look at where it binds, you can see it binds right on the same site that the nicotinic acetylcholine receptor binds, which suggested to us that it would be neutralizing, and explains how it's so broad, because those are the positions that the snakes have never managed to mutate, or their toxin doesn't work anymore. And so this sort of molecular mimicry of the conservation site was what we're looking for, and that's exactly what we found. I know I'm getting a little short on time. This is the last couple of slides. I'll go a little light here. The punchline is we took that toxin and we put it into mice. We found the one antibody provided complete protection from a lethal challenge by the neurotoxin from black mamba, coastal taipan, Cape cobra, common krait. This is spanning multiple continents and great phylogenetic diversity. We then went to whole venom, and we found for some snakes that single antibody was enough to provide protection from the whole venom. That was 4 species of cobra spanning multiple continents. It was the king cobra, which is actually not a cobra. It's so named because it eats cobras, and the black mamba. Those were all entirely protected by a single antibody, which tells you that the venom that they're giving you is the dominant thing that's killing you is the neurotoxin, and we're now protecting it. There's some other stuff in there you don't like, but that's what's killing the mice at this doses. There were other venoms where our antibody helped a little bit, but it wasn't enough. And we put a second agent in there, which is called varisplatib. It's a PLA2 inhibitor. And now with 2 agents, we were able to provide complete protection to the tiger snakes, the inland taipan, the coastal taipan. And it seems like the PLA2 was more of the driver in those venoms. And then we had a synergy effect, but the animals are still dying when it comes to the common krait. You can see either drug alone was only partially protective. When we add them together, they have a great extension of life, but they're still dying. So there's a third agent in there to complete universality with respect to that venom. And we're working on it. And then there's some where we think that that third agent actually was the driver. And so things like yellow lip sea krait and eastern brown, it helps a little bit what we're giving them, but it's this third thing. And we think it's the short neurotoxins, or they're cytotoxic short neurotoxins that are actually driving lethality. And so, but the punchline is pretty remarkable. If you look at the phylogeny of all the Elapidae and the geography, we've managed to take 2 agents and protect against the majority of elapid snakes throughout the globe of the continents that have them. And with the completion of the third agent, we think we're going to achieve coverage. There's a separate set of a couple antibodies we need for the vipers, but the point I want to leave you with is that broadly neutralizing antibodies could dramatically improve the quality of antivenom globally. And the problem was not nearly as hard as it initially appeared, because nature's lazy. Even though there's 600 snakes and they all have lots of venom, they all use the same protein fold. So it's really just 10 toxin classes across all snakes, and 5 of those aren't going to kill you. They're not going to mess your leg up. It's the other 5, is these top 5 you have to target. So in principle, 5 antibodies, if each one was perfect bnAb, would create a universal antivenom. That's the hypothesis. We're working on it. We're partway there already, and you can check out the paper. So that's the talk I wanted to give today. I'd like to stop now and ask any questions that came up. Hopefully I've convinced you of the power, importance, the engineering practicality, and the vaccine opportunities of broadly neutralizing antibodies. And again, I'd like to thank Sino Biological for their help literally across every one of those projects as I've been advancing them.
Thank you, Dr. Glanville. That was a really, really interesting talk. Wonderful. One of the best that we've had thus far for the Lock and Key webinar series. And in the interest of time, I will go ahead and jump right into the questions. And the first question written into the chat box here is from Tilahun Yilma. And Tilahun asks, protection against many viruses depends on inducing CMI, not antibody responses. A good example of this is measles. Are you ignoring CMI in your system for protection against viruses?
So I disagree with that. It's been established with, so first off, I would point you to the fact that broadly neutralizing or any antibody therapies actually work and they provide protection. We know that for the coronavirus. We know that adoptive transfer experiments work for flu, and there are a number of other pathogens where this is the case. There are some pathogens where you have to consider other aspects of immunity, but I think you just have to ask, hey, when we put antibodies in through an adoptive transfer, did it protect? Or if we give an antibody therapy, did it protect? If the answer is yes there, then you have to back off that point. And the answer is yes. So I don't agree with your point. I think of course, there are other types of immunity that matter. T-cell immunity matters in some cases, but I'll point out for flu, they tried a T-cell specific epitope method and it did not work by itself. That was the BioNvax. It was adjunct, but it was not standalone. And so that would be my answer to that point. I'm happy to receive a follow-up if I misunderstood the question.
Great. Thank you, Dr. Glanville. Appreciate that. I guess you and Tilahun will agree to disagree on that point. I knew we were going to get a lot of snakes questions because that was incredibly fascinating. Yeah. Let's see, the name here is Carlos Ruben Ortega, I believe, is asking, were any American snakes tested, or was it all kind of the more exotic ones, I guess?
Yeah, it's a great question. So the long neurotoxin exists in the elapids. So the snakes broadly separate into the Viperidae, the vipers, and the elapids. And the New World doesn't have that many elapids. We have the coral snake. And so we are testing it. It was harder for us to get the venom for the initial experiment. So it was a technical reason we didn't have access, which was annoying to me because I was applying for NIH grants. So I wanted to show. We have since, we're working now on Viper targets, the PLA2 Viper target and SVSP. And so for those, of course, yeah, we have a lot of New World Vipers. The way global geography works is the New World is mostly Vipers. Australia is entirely elapids, and the rest of the world is a mixture of both. And so we do work on them, but this current study is under analyzing New World snakes because it's only the coral snake.
Got it. Coral snake venom is quite difficult to obtain, I'm assuming, right?
Yeah, we went to there's these Venom Week, and there was a Gordon Venom conference, and we found some collaborators that have them in Mexico, so we'll be working with them.
Yeah, coral snake is very prevalent in Texas too, which is where I am. We have to be quite careful of that snake, especially in East Texas where it's quite humid.
Yeah.
So Dipali Rawat asks this question. I'm not sure I understand it, but I'll give it a shot here. How are the broadly neutralizing antibodies different from more classical polyclonal antibodies?
Yeah, sure. So the difference is, I think I'll show you two examples. One is anti-venom and for vaccine. So when you get a vaccine, you're getting polyclonal antibody response. So it's a bunch of antibodies, but unfortunately, the majority of them are not going to be against conserved sites. So the conserved epitope is rare. And so your polyclonal is almost entirely not against the conserved site. That's why they have to change the flu shot every year. A broadly neutralizing antibody is targeting the conserved site. If you could create polyclonal broadly neutralizing, that's what our technology does, then you get the best of both worlds. And in general, I don't like putting all my eggs in one basket. I think more antibodies is better. But traditionally, with current immunization, when you go polyclonal, you're not going broad. And that's been the whole problem with broad spectrum vaccine design. Similarly, with anti-venom, the current polyclonal mixtures are a bunch of antibodies, but they're against whatever the horse or the sheep was immunized with. And so they are going to be overfit or overly specific. The majority of them are going to recognize epitope determinants that are specific to the snake venom that they were immunized against. And some of them will cross out a bit to other snakes. Versus a broadly neutralizing antibody, we've picked out that 1 in a million antibody on purpose that recognizes the best site, and rather than dilute it amongst a bunch of other antibodies, let's just make the whole product that. So it ultra focuses and it's ultra broad to be able to dedicate the drug to the broadest immune protection. And in the case of antivenom, that results in a superior outcome.
Great. Just going right down the line then, Colin Hebert asked a technical question here regarding the reagent testing for neutralization assays. Did you consider a label-free method for measuring viral neutralization?
I'm sorry, could you repeat the question?
Did you consider a label-free method for measuring viral neutralization? Is that relevant to your technique?
I'm just trying to contact me, please. I'd be interested in hearing more. I think that's my correct answer. So no, we did anti-MP, but if you're familiar with alternatives, I'd be interested in hearing them and understanding what the advantages are.
Yeah, no worries.
I'd like to add one point to the first speaker, because I may not have given their question full credit, benefit of the doubt. So we do look at T-cell immunity. We believe that our technology also focuses towards more conserved T-cell epitopes. And of course, T follicular helper cells are important for stimulating the antibody response. So we know for flu that antibodies drive the dominant need for protection. And that's been demonstrated by the failure of BioNVac and by a number of studies of selective ablation and adoptive transfer. So I don't think there's any room to argue against that for flu, but to your point, yes, we actually do look at the T cell compartment quite a bit because we believe that our technology is focusing the immune response towards conserved T cell epitopes that are shared throughout our variations, because they're also at higher concentration if it's conserved than the sites which are not conserved. And this is something we're still characterizing.
I think so. Good. Yeah. And next question. This was actually a question I had. This is from Alejandro Allegon, and I know you've got a paper out on this, it's in process, but can you give us a status update on the snake universal anti-venom and where it stands and when we might have access to it?
Sure. Drug development takes a while. So the paper shows that the paper focuses on the first of the 5 toxins. And so that's the long neurotoxin. As it stands, that could be a product against like all cobras and some other snakes. It's expensive and it's a neglected disease area. So I don't think I want to go do a bunch of different clinical studies. I'd rather get it right first. And so we already have more progress in preparation for 2 of the additional toxins. And then there's 2 that we're still working on. It's fair, we're early on. So I estimate about 18 months to complete the complete product. At that point, we would be manufacturing. GMP takes a while. That's at least 12 months. And my experience is that there are delays. After that, you're talking about a 6-month phase 1 study. That's a pretty clean dose escalation study. And these came from a human and he's fine. So we suspect they'll go well. After that, it's difficult to say, but roughly 1.5 years, maybe 2 years. It depends on recruitment. We'd be having recruitment across, 6 or 8 sites globally. And on the completion of that, then we would have the drug available. So that takes you out to maybe 2028 to 2030 when the drug would be available on market.
Sounds good. I'm a hiker, and I can't tell you how many times I've stepped on a rattlesnake or a copperhead, and especially up in the Appalachian Mountains. I'd love to have that in my backpack. So
that's the other advantage is that current antivenom, they have to give it IV because it's super immunogenic. It's horse or sheep antibodies, and it's polyclonal. So a lot of it's not against the target. Actually, 70% is against unrelated. But it's going to cause an immune reaction when you try to ameliorate that going IV versus ours is fully human, and we can modulate the effector function response to make it even safer. And that means you can concentrate it. You could deliver it as we call it a ven pen, like an EpiPen, but with venom antibodies. So it could be delivered on the site rather than having to wait and have tissue damage as you're transported to a medical center.
Sounds great. I think we've got time for one more question. The rest of the questions we will get to remotely via email, if that's all right. This question, I'm just going down the line here, not playing favorites. It's from Benedict Offer, and it is, did the study of vaccine that targets alpha neurotoxin epitope take into account variations that occurs from snakes from different geographical locations?
Yeah, that's an awesome question. So there's this known problem that current antivenom, and actually, the current antivenom for vipers works better in one part of Arizona than another part of Arizona and in Southern California. And it comes down to the fact that even though you call them all Western Diamondback, there's genetic polymorphism and variation in the species. That is a huge argument for broadly neutralizing antibodies, because if there's variation in a species, then the specific snake, not just the species you inject into a horse or a sheep, is going to impact the behavior of your drug. Versus if you go for broadly neutralizing antibodies, if it's able to span 100 million years of evolution, then it damn well is going to be able to handle the last 40 years or whatever, 500,000 years of regional allopatry and local polymorphism. And so that's another argument for bnAbs, is that they're hitting a site that evolution hasn't managed to modify in 100 million years. And so it's very safe from local polymorphism.
Sounds great. Well, with that, Dr. Glanville, we'll go ahead and wrap up. First and foremost, I want to thank all of the attendees. We had one of the biggest turnouts ever for this very interesting talk. Just very quickly, I have to run through the countries from which folks attended. Nigeria, of course, all across the United States, Germany, Serbia, Ghana, Mexico, Pakistan, Kuwait, Burkina Faso, Kurdistan, Cyprus, Egypt, Algeria, South Africa, Greece, Canada, Taiwan, Iraq, Saudi Arabia, and Benin. And so thank you all for attending. We appreciate the big turnout and the excellent questions. The ones that I didn't get to, I apologize. We will get to those offline. And Dr. Glanville, I want to thank you as our speaker. Again, it was one of the most interesting and I think very, very promising seminars that we've had to date. Let me congratulate you on your success with distributed bio and with Centivax, work with Mark Davis. And also we appreciate the plugs at Sino. Thank you very much for that. That was very nice of you. And talk to me offline about getting those cell lines. All right, I'll do my best for you on that.
All right, outstanding. You know, I don't do plugs if I don't mean it. I really did mean it. Like I was not able to start that flu work if it wasn't, because I wasn't going to able to afford it is what it was. If I had to go, it was that starting barrier where I had this cool idea and nobody heard me. And also they're like, I didn't have a PhD yet. I was working on it still. And so I suddenly was like, wait, they have an inventory. I can actually text the hypothesis out. And then I built a facility in Guatemala. I was like literally willing to shovel through pig shit, which I did in order to test this hypothesis, but I would have been stuck if it weren't for Sino. And they just come over and over again with the coronavirus research we do, with the antibodies we're engineering, with some of these new constructs for HIV and the coronavirus and other pathogens. They've been a great partner. So I meant what I said, and I want to thank everybody for your time and attention. And I had a lot of fun. So thanks.
Appreciate that. Appreciate that. World's largest selection of viral antigens and corresponding antibodies. That's my plug for science. I had to do that. Dr. Glanville. And one more person, Christine Lay, who works with Sino in our marketing department and also in sales a little bit. She organized and executed this webinar. So Christine, thank you for all your efforts as well. And I bid everybody a good evening, good night, or good afternoon, wherever you may be. Thank you.
Cool. Cheers.